Thanks for everybody who came out. Some of you saw a version of this a couple weeks ago, I think. This is the fourth time I've given this talk in the last month. So I'm covering all corners of the campus. So plant diversity is amazing. From unicells to giant sequoias, plants span over 21 orders of magnitude in mass. They're found in virtually every habitat where liquid water and light are available, and their metabolic and morphological diversity rivals that of any other clade. And my work has been governed by a single overarching question for some time now: is can the scaling of plant form and function be described by general models based on physical first principles? And by first principles, we mean starting at established laws of physics and then building up from there. We think the laws of physics operate most everywhere in the known universe, and so we should be able to take those physical laws and construct plant models from them. And so this usually involves some sort of mapping between plant form, plant geometry, and function, rates of material flux. So today I'll give a little background, a scaling primer for those of you who aren't used to thinking in this allometric space. Talk about some of the early work that catalyzed a lot of interest in this area. I'll introduce the metabolic theory of ecology, and then some work I've done evaluating and extending that body of work, and then some more recent work moving away from that earlier framework into some novel approaches. So the first attempt that I'm aware of to predict form and function in a scaling context comes from Galileo in his dialogues concerning two new sciences. And Galileo was interested in why the limb bones of large mammals were proportionally thicker than the limb bones of small mammals. And he reasoned, okay, if surface area is proportional to length squared and volume is proportional to length cubed, then if we look at the relationship between the two, you should have a scaling exponent of 2/3. Today we would log transform that. There are valid statistical reasons to do that, and that has the effect of linearizing this relationship, which allows you to compare empirical slopes between groups or compare empirical slopes between a model and data. I often use slope and exponent interchangeably. One is on the linear scale, one is on the log scale. I'm talking about the same thing. And so the model that Galileo described is geometric similarity, and it simply says for a self-similar isometric object like a sphere, we have these expected scaling relationships between things like length and diameter, which for a sphere are isometric, the same thing, and volume. Now, if density is constant, then we can equate volume and mass. Are organisms geometrically similar? Often not. And so it begs the question, what alternatives do we have? So a little background. Back in 1932, Max Kleiber published a paper in the journal Hilgardia, and he was interested in how metabolic rate scaled with mass across organisms of different size, and this is the figure from his paper. He really had just a handful of data, and he actually used a slide rule to get the slope for this relationship, and found a value of 3/4. Now, this caught some people's attention because prior to that, people had thought that this surface area to volume argument based on geometry, for endotherms, the idea was that the rate at which we can dissipate heat should be related to our surface area, and then if we scale that with volume, it should be something based on 2/3. So the fact that it was 3/4 caught people's attention, and then the identification in subsequent groups of this recurrent 3/4 relationship, combined with the publication in the 80s of several compendia of allometric relationships, these three books by Robert Henry Peters, Knut Schmidt-Nielsen, and Bill Calder. If you've never looked at these books, they're rich with these allometric relationships. And what they find is that a lot of the scaling exponents, particularly when we're looking across species, most of this work was done on mammals and birds, and when we look across species, the scaling exponents often are some multiple of 1/4. Things like heartbeat frequency, aorta radius, blood circulation time, population density across species, the scaling exponents are all some multiple of 1/4, and this becomes known as the mystery of quarter power scaling. in biology. So a lot of people have focused on this core metabolism to mass relationship because it underpins lots of other things. For example, biological rate. So if we take our metabolic rate as proportional to mass to the 3/4, if we divide both sides by mass, mass specific rate is proportional to mass to the -1/4. And so things that are tied to mass specific metabolic rate, like heart rate, development rate, mortality rate, are expected to scale and have been shown empirically to scale with scaling exponents that are some multiple of -1/4, not some multiple, that multiple. Here's an example from Brown et al. In Ecology 2004, turnover rate in different plant communities as a function of the average mass. The slope is very close to the expected -2.5. Biological rate times are the reciprocal of biological rates. So things that are related to time, this is a figure from Lindstedt and Calder, 1981. Calder worked mostly on birds. All of these different aspects of bird physiology across species, muscle contraction, cardiac cycle, circulation of blood volume, incubation period, growth, lifespan, all have slopes that are close to 1/4. And so, as you can see, that core relationship between metabolism and mass underpins all of these scaling relationships, biological rates, biological times. And so where does this all come from? What's the answer? So in 1997, Jeff West, Jim Brown, and Brian Enquist proposed a model that they claimed explained the origin of these quarter power scaling exponents. The first paper in Science was a treatment for the mammalian cardiovascular system and respiratory system, a suite of predictions coupled with data where it was available. It's rare in ecology and physiology to make so many predictions from a single model, and so I think this is one of the reasons that this body of work got so much attention. They followed it up in '99 with a treatment for plants. Again, lots of predictions for different aspects of plant morphology and physiology as a function of plant mass or branch radius. Jamie Gillooly was a postdoc of Jim Brown's, and he said, hey, temperature is important, and it explains the offset between these different groups. Let's bring that in. And so this collectively starts to become known as metabolic scaling theory, metabolic theory of ecology, various acronyms. But the core idea is that size and temperature explain an awful lot about the variability in metabolic rate across these different life forms. And the crux of their model, the idea, which is, I think, satisfying intuitively, is that energy minimization has resulted in an optimal fractal network. So our heart has to work to deliver blood to the capillaries. The idea is that over time, natural selection should have honed the network geometry to minimize the resistance to flow, because you don't want your heart to work any harder than it has to. And so I think that idea is satisfying. It turns out that finding the optimal geometry of that network is a much trickier problem than it has been presented. I'm not going to go into all of the details of that today, but we've written about it in some of the papers that I'll mention. But the basic idea of their model is that we have this fractal network. It's self-similar across scales, and it can be described by three terms: the branch lengths ratio, which is the length of daughter branch to parent branch; the branch radius ratio, which is the radius of daughter branch to parent branch; and the number of daughter branches per parent branch, or the branching ratio. And what they did was they solved for the geometry that, again, minimized resistance to flow. And what they claimed was that a network that exhibited volume-filling branching, and you can think of this three as kind of the three-dimensional volume that the network fills, and area-preserving branching. So if I have a conduit that branches into two daughter branches, the cross-sectional of the area of the daughter branches should equal that of the parent branch. Area-preserving branching is also velocity-preserving branching. And so they argue that this led to energy minimization and why we see these quarter power scaling exponents. So for plants, you can imagine their model like this. So you have this fractal generator that grows, your trunk gets bigger. Because you know the geometry, you know the volume. If density is constant, then you know the mass. As this thing grows, you know the volume and you know the mass. They argued that surface area should be proportional to the number of branch tips. You can imagine there's an idealized leaf on the ends of each one of these branch tips. And if the size and properties of those leaves don't change across species, so they have an average size and an average physiology that doesn't change across species, then if photosynthetic rate or metabolism is proportional to leaf mass or leaf surface area, then you can link surface area to volume. And that's the basics of their argument. So does the data support the theory? Well, these guys were in the comfortable position where there was already a lot of data for quarter power scaling as a central tendency across these different groups. We had a lot more data for mammals and birds than we had, but subsequently we've collected a lot of data for plants. However, since the publication of these papers, we've looked at some groups in more detail. This is a figure from Brown et al. In Ecology 2004. Lots of different groups, temperature-corrected biomass production rates over, I don't know, 30, 40, 50 orders of magnitude in mass. The empirical slope is very close to the predicted 3/4. Lots more data on plants since the publication. Mines looked at water flux in trees. There's different proxies for metabolism in plants. Water flux is one, dark respiration is another. He looked at water flux and found that it did in fact scale close to the expected 3/4. Reich et al. Looked at small plants. These were mostly seedlings and saplings and found an exponent of 1. Moray et al. Looked at this is the largest data set of which I'm aware, and they found that it was somewhat nonlinear, going to close to 3/4 up here, but closer to 1 down here. There's reasons to expect that it might be 1 down here. A lot of small plants, all tissue is metabolically active, and so you might expect then metabolism to be just strictly proportional to mass. But then as they get larger, these surface area arguments, a lot of the tissue of a large tree is not metabolically active, and so it's easier to conceive that it might be surface area limited at that point and then may converge to some 3/4 scaling. So a lot of what happened following the publication of these models is that people would go out to their organism or their system and they would measure these empirical exponents. They would compare them to the predictions of the model, and then they'd say, okay, it supports or doesn't support the model. And that's okay, except that I and some others felt that there are some earlier steps that we really needed to kind of evaluate more critically before we spent so much time down here, and we presented this in a paper a few years back in Ecology Letters. First, is the math correct? If the math is fatally flawed, there's not a lot of point in exploring the subsequent predictions. If the math is robust or has minor errors, then we can proceed. Are the assumptions okay? Are plant networks fractals? Are the simplifying assumptions okay? If the assumptions are so far disconnected from reality, then it makes it hard to make that model work. And then you can start to look at the predictions and the extended predictions, of which there are many. And I have spent a lot of my work at the level of these assumptions. Are plant networks fractals, and are the simplifying assumptions okay? So we'll look at these. Now, I have long felt that the West-Brown-Enquist model was an incomplete or insufficient characterization of the way plants work for a number of reasons. But I still think that there's a lot of value in evaluating these assumptions because they can be used to underpin further work. And it's just good to know from a first, a basic science standpoint what the answer to some of these questions are. And so we'll go at them one or two at a time. The plant has a large number of orders of branching. The network is volume filling such that each branch has an associated service volume. So at the time I was in graduate student Brian's lab, and I'm reading these papers and looking at these assumptions of large numbers of orders of branching and volume filling, and then I'm looking at the plants in the Sonoran Desert thinking I'm not really seeing it, Many of these plants branch not at all or minimally, and so I got into the details of their model and started playing around and said, okay, if we relax that assumption and say our plant, this would be a caricature of a West-Brown-Enquist plant where the internal vessels parallel the external branching morphology, and that's fairly true for a lot of trees. But cacti are doing something more like this, where you have this reduced surface area that's minimally branched, but has a highly branched internal vasculature, and that's in fact what cacti do. And so when you do that, you come up with a different set of predictions from the original fractal model for different aspects of plant morphology: height, diameter, mass, canopy spread, water mass, and the empirical slopes were much closer to the minimal model than the fractal or geometric similarity. And so this was encouraging. This version of the model still predicts that surface area scales to the 3/4 power volume because that internal vasculature is still highly branched. And in fact, when we looked, I collected data from the literature, and for these 3 different species, the scaling of surface area to volume is actually very close to 3/4. So it encouraged me to push this approach further, and so what we said was that instead of assuming that this lengths ratio is always a number, n to the 1/3, let's just represent it by this parameter b, which is the branch lengths ratio, which allows that to vary. Similarly with the radius ratio, we'll represent that with a, and that's our branch radius ratio. So instead of predicting a single number for the scaling exponents for these different relationships between basal stem radius and mass, length and radius, we predict they're all going to be function of A and B, which can describe the geometry of a given species, but can vary between species. And so these aren't free parameters that take on an unlimited range of values. In fact, we think that they'll be fairly constrained. We can envision one end of the continuum area preserving branching, which is velocity preserving. At this end of the continuum, area increasing, velocity decreasing. This might allow things to slow down to allow exchange across boundaries. An example of this would be Murray's law. In fact, in the mammalian cardiovascular system, what you see coming out of the heart is area preserving branching. But then when you get out to the capillaries, it converges to something called Murray's law, which is area increasing and allows things to slow down to allow exchange. The branch lengths ratio continuum, at this end we would expect volume filling branching, think 3-dimensional things. Down here, more 1-dimensional. It's obviously a 3-dimensional thing, but it's investing most of its mass in growing in 1 dimension. And this can be thought of as the dimension of the volume that the network fills. So I scoured the literature for empirical measurements of these variables, and there are some, and they're consistent with the range of values that we can expect. But it was clear that we needed many more empirical measurements of actual networks so we can see if they're actually behaving in the way that we are predicting. So because we don't predict a single number for the scaling exponent, like you would see in West-Brown-Enquist or an elastic similarity, we predict that that scaling exponent is going to be a function of the network geometry as described by A and B, which describe the lengths ratio and the radius ratio. So to evaluate this, what we can do is look at the covariation between exponents. And so here is a black line, which is the predicted covariation function between these two exponents. For example, West-Brown-Enquist would say A is 1/2, B is 1/3, which would give you a value for radius to mass of 3/8, length to radius 2/3, and so that becomes a point on this continuum. So we can then compare empirical data to these predictions, and that's what we're looking at here. So the black line in all 3 of these is the predicted function. It's a continuum which represents all hierarchical fractal networks. Each point here represents the scaling exponent from a species. So for example, this is a woody plant. Either I or someone else collected 20 to 30 individuals of that woody plant species, from the smallest one we could find to the largest one that we could find, measured its basal stem radius and mass, its length and radius, and then that exponent is the observed exponent for that species. And so we have lots of data here. Down at this end of the continuum, where we expect things with area preserving branching that are 3-dimensional, is where we find the woody plants, the succulents, perennial and annual herbs tend to cluster down here. Now, as we get out here, getting closer to the area increasing one-dimensional end, we get the green dots, which are angiosperm leaves. They're more two-dimensional. Kind of makes sense. They're kind of flattish. And then the black points are angiosperm leaves, which are conifer needles, which tend to be more one-dimensional. And so this approach explains and tends to capture fairly well the curvature of the data for these different scaling exponents across whole plants and leaves, and does a much better job than the single value that emerges from West, Brown, Enquist. I've also plotted the expectations for stress similarity and geometric similarity, which fall along this continuum. The large circles are the empirical means from the different groups. And what this is really telling us is that morphology and allometry are linked. Shape matters, and I think this is intuitive. If you have something that's highly branched, it's going to increase in mass slower, increase with mass in height slower than something that is investing most of its mass in growing up. And so this allows us to link those two things. We've also looked at patterns of biomass partitioning within this framework. So here we're looking at the height of a plant versus its leaf mass exponent, length versus stem mass, leaf mass versus stem mass. Again, each point represents an empirical exponent from a species. The prediction from the model is a surface, and so I've superimposed two lines. This first is holding the lengths ratio constant and allowing the radius ratio to vary. The second is holding the radius ratio constant and allowing the lengths ratio to vary. The intersection of these two lines is the expectation from the West, Brown, Enquist model. And what you can see is that most of the variability is falling out along this lengths ratio axis. Now, if we think of plants in the context of Poisson flow, this is the model that everybody uses to model resistance in plants and animal networks, tubular networks. And it says that resistance is proportional to length to the power 1, but radius to the power 4. And so because changes in radius have such a large impact on resistance, we argued that length is a much more plastic trait, both developmentally and evolutionarily, that you can change your length without much cost in terms of resistance if you're a plant. And anybody who's done light experiments on plants has seen this. You know, if you deprive them of light, they become kind of leggy, they get long. And so that's why we think we see much more variation along this axis. We've also used this to kind of constrain the state space where we think leaf scaling exponents will occur. So this, again, each point represents the empirical exponent for a leaf for a given species. I collected leaves 20 to 30 from the smallest leaf I could find to the largest leaf I could find, figured out the exponent, and then that becomes a data point on this plot. Surface area to radius, length to surface area. I won't go through both of these, but what we then do is put the expected lines for a two-dimensional case, a three-dimensional case, and area-preserving branching, and everything tends to fall between these kind of constraint lines, with the exception of this point out here, which is Ginkgo biloba, which is an ancient gymnosperm. It lacks hierarchical branching, so I'm not sure what's going on there. Okay, so we've looked at some of the predictions and how they change if we modify this network geometry. Another assumption of the model is that branches are elastically self-similar. This is McMahon's elastic similarity model, published back in the 70s. It says that the ratio of a branch's length to its deflection remains constant. They're elastically self-similar. They have similar elastic properties. People have looked at this in detail. I'm not going to spend much time on it, but the model predicts the length. This is an idealized column. The model is a column, but people have used data from the height of trees to test the model. We've done that here, height and diameter. The empirical relationship for angiosperms is close to the expected 2/3. Gymnosperms, not so much, and when you look across the groups, it's somewhere intermediate. So elastic similarity has had some empirical support, but I think it's kind of limited in its scope. Another assumption is that bulk tissue density is constant for all tissues and invariant with plant size. It's not. We've looked at that across species. Actually, large things have lower density wood on average. And in this paper, we go on to explain that plants achieve most of their increase in height through changes in geometry, not changes in wood density or wood strength. In fact, wood density and wood strength don't vary much as a function of plant size, or density actually decreases. Branches are terete cylinders. I think that's this is like the easiest one. They're fairly cylindrical. I think most of us would accept that as being fairly valid, at least a good place to start. The structure and function of the terminal units of the network, petioles, leaves, don't vary as a function of plant size. So I asked Ian Wright to use the GlopNet data set. And so we looked at leaf mass per area, leaf lifespan, leaf area, nitrogen, phosphorus, and photosynthetic rate on a mass or an area basis. And for all of these relationships, while there's considerable variability, that variability is not explained by plant height. So large plants don't explain the variability in these leaf functional traits, with the exception of leaf area. There's a modest trend with leaf area. So large trees have, on average, larger leaves than small trees. And so depending on and we go to length in this paper to discuss what is meant by invariant. It's something that has come out of the mathematics literature that biologists have co-opted without often explaining what they mean by that. And so depending on how comfortable you are, you might be willing to accept the assumption that leaves are invariant across plants as a function of size. Okay, arguably the most important assumption is that the network is strictly hierarchical and self-similar. So is it a fractal? Is it the network? And so I remember thinking we needed lots of network data, but trees are very hard to measure. But leaves might be easier, so I spent a good portion of my postdoc developing software that allows us to take an image of a cleared leaf and extract the geometry and topology of that network. This is a screenshot of what the software looks like. I'm happy to demonstrate it to anybody, but what we've done then is examined lots of different leaf networks. And so a fractal model predicts a power law distribution of vein lengths and vein diameters. So we looked at this across 353 species. We took the leaf, we measured the geometry of the network, and then we can look at the frequency distribution of, we called it edges in this paper. It's a vein. We called it edges because we used lots of graph language in this paper. It's a vein length. And so the frequency distribution of vein lengths and vein diameters to see if they are in fact both power laws. And we found that for diameters, they're all fairly well approximated by a straight line on a log-log plot consistent with a power law. But for lengths, they were linear on a log linear plot, not a power law, and in fact better fit by an exponential model. And so leaf networks are in fact this mixture of distributions, power law distribution of diameters, exponential distribution of lengths, and this is the same pattern that's found in river networks, and which I find fascinating, but they're kind of doing something very similar in that they have an average basin area over which they start to drain. That sets this characteristic length scale of the network. Leaves also have diffusion. There's a length scale over which diffusion is no longer a good way of getting fluid around, and then you need bulk flow. That's why we evolved networks in the first place. And we argued that diffusion limits set that characteristic scale of the network, that average length scale. And so this is the exponential model. P is the inverse scale parameter, which we equate with the average length of that network. And so leaves have this characteristic length scale. And so when we look at the average vein length, or the average distance to the nearest vein, or network density, across leaves of different size, we see that that average length scale doesn't change. And so big leaves, even though there's variability, again, it's not explained as a function of leaf size. I've also looked at the geometry of the networks as a function of where you are in the network. And so Here I've got the side lobe of a fern leaf because it's simple and easy to see and to understand what I'm explaining. So we take the image, threshold it, apply some of the algorithms, and then I've shown a strictly hierarchical network here for illustrative purposes. Leaf networks are reticulate, they're loopy, and so to find we wanted to apply a ordering algorithm to look at how branch order affected geometry. To do that on a loopy network, what we did is we found the minimum spanning tree. So we have a network that connects all of these nodes. We find the minimum spanning tree which connects all nodes without forming loops, and what we do is we minimize for radius to the power 4 because it's proportional to conductance in that Poiseuille model that I talked about. And that basically captures the large veins and that hierarchical part of the leaf vein network that you see, but then it's like cutting the small bits that then make it loopy. And then we can look at how geometry changes as a function of order by applying a Horton-Strahler ordering algorithm that comes out of the river network literature. So Horton-Strahler starts at the tips, they're order 1. When two 1s come together, that becomes order 2. When two 2s come together, that becomes order 3. And so then we can look at how geometry changes based on order. And what we found that veins of order 1, again, that's the smallest veins in the network, they have a mean that's close to what we expect under Murray's law. I mentioned Murray's law earlier. This is a paper from 1926, C.D. Murray. He was a physiologist, and his model is based on minimizing resistance to flow and minimizing maintenance cost of blood volume. And it predicts that the sum of the radii cubed remains constant, not the sum of the radii squared, as in area-preserving branching. It's been shown to be true in capillaries and in leaves, also obey this Murray's law scaling, but when you get up to the larger veins, they actually shift to being closer to area-preserving. This is the same transition as observed in the cardiovascular network that I mentioned earlier. Leaving the heart, it's area preserving. Going out to the capillaries, it converges on Murray's law, which I find super cool, with the caveat that these are vessel bundles, not xylem. And so it's a bundle, a leaf vein is a bundle with xylem in them. There's reason to believe that the internal network geometry parallels the external network geometry in leaf veins. We've tried to evaluate that empirically, but it's actually very hard to section some of those small veins, and so still working on that. Okay, moving forward. So I felt that there was enough wrong with the West-Brown-Enquist model that we needed to start to entertain alternatives, and lots of people have felt that way for a while. And I had a student, Sarah J. Knox, who was actually investigating leafing intensity. Leafing intensity is the number of leaves per stem volume in plants. In the Banksia genus, which is an iconic Australian genus, most species endemic to Australia, and they have this great diversity of leaf form. And one day I was looking at her data, and I just, on a whim, plotted stem surface area to stem volume, and noticed that the slope was close to 3/4. Now, the West-Brown-Enquist model assumes elastic similarity, and in their model, surface area is these leaves that occur at branch tips, not the stem surface area, okay? And so the fact that this elastic similarity, in fact, would predict 0.625 for this relationship. So the fact that this was 3/4 obviously caught my attention, and so I decided to push it a little further. And to make a long story short, if you take the Hagen-Poiseuille equation, and some minimal assumptions, many of which underpin other approaches. Volume conservation, people don't generally have a problem with that. Constant viscosity, don't have a problem with that. Velocity preservation, that's also been a feature of other models, area preserving branching. And then a constant pressure drop, which we don't have a lot of data on. It doesn't say that pressure is constant, it just says that the drop in pressure is constant. This is tightly constrained version of the model. You get the same predictions with what I call a loosely constrained version of the model, where all you need is volume conservation and area preserving branching. What you lose is what drives that area preserving branching, with the velocity conservation being what drives area preserving branching. But with volume conservation and area preserving branching, you still get these same set of predictions. And what it predicts is that your network will have a length to radius scaling going to the power 2 and a surface area to volume scaling going 3/4. Michael Renton, who has a background in mathematics, helped me a lot. When you get into summing of the series of things with side branches, it can get a little tricky, and so he was very helpful in making my clumsier math look pretty. And so to contrast these two approaches, this is elastic similarity, or West Brown-Enquist. They have this idealized fractal plant with a leaf at each tip. What we've said is that leaves are in fact arrayed along branches, and that's what plants do. So it's more consistent with biological reality. And so for a cylinder, and this is basically a cylinder that we're modeling, we can look at its length, diameter, surface area, and volume. And for those 4 things, there are 6 pairwise ways that we can look at them: length to diameter, surface area to volume, diameter to volume, length to volume, diameter to surface area, length to surface area. And so focusing here on the length to diameter relationship, we can represent it with this parameter alpha. Now if alpha is 2, then all of these other relationships fall out based on these formulae. And that's under what we called flow similarity. Similarity in volume and velocity flow. I can get into the criteria for similitude and whether or not this obeys all of those if anybody wants to. Now if alpha is 2/3, that's elastic similarity, and we get different predictions. What we argue in this paper is that different parts of the network might be doing different things. I've shown that in leaves. The mammalian cardiovascular system does that. And what we argue is that at branch tips and for small plants, it may be that hydraulics is more important than biomechanics, and so the geometry is going to be more driven by the flow constraints. Whereas if we're down here at the base, a tree has to support a lot of mass distal to that basal branch and these early branches, and that we might expect elastic similarity to operate more down here. When we do that, we predict that the exponent will change as a function of where you are in the network, or if we're looking across plants, as a function of the size of the plant. And we can't predict the exact function because it's going to depend on the specifics of the architecture, but we can predict that it will be concave or convex. And I mentioned earlier that people have evaluated elastic similarity by looking at the height of the plant and its diameter, but tree height and path length are very different things, and I think that becomes very important here, and I'll show you why in a minute. We have lacked an alternative to the West Brown-Enquist approach. It's been a real problem in the field that there's been this one model but nothing to compare it to, and so here this is an alternative that the scope of which I don't know, but the early data are encouraging. And this, I love this title of T.C. Chamberlain's paper. This goes back in 1890, The Method of Multiple Working Hypotheses. With this method, the dangers of parental affection for a favorite theory can be circumvented. And I think parental affection for theories is a danger. And so, you know, we generate more than one hypothesis and test both of them or multiple at the same time. So it assumes area preserving branching, velocity preserving branching, and so I've evaluated that in tree branches and leaf veins, and we find the empirical mean to be very close to the expectation of 1, daughter branch area over parent branch area equals 1. Maximum sap velocity, this data is harder to come by, but Savage et al. Looked at this in 2010 and they found that it didn't vary much, not statistically and not as a function of branch radius. Now, if you're within species and the range is small, you do get some non-zero relationships there, but it's not a strong relationship across species. I said that we would predict, looking across large data sets, that height and diameter would be concave, height and mass concave, diameter and mass convex. This is lots of data that I've collected myself or collected from the literature, thousands of data points, and the curvature is correct in all 3 of these cases. And there's lots of published plant height to diameter relationships, and they all, for large data sets, generally all look like this. So focusing on this length to radius squared relationship. So if you go out and you collect data, these are data for eucalypt trees. We measured the geometry and topology of 20 eucalypt trees getting up to about this big. It's really hard to get this data. If you just plot length versus diameter for each internode, you measure the length and diameter of every internode in that tree, and then you plot them, what you often get is something like this. It's a shotgun blast. And I think this is kind of has obscured this underlying pattern. Now, if instead I look at the subtrees within a tree, so for every internode, I measure its diameter, and then I look at the dimensions of everything distal to that point, the length distal to that point, the volume distal to that point, surface area distal to that point. The data then collapses around a mean that's very close to 2, which I'll show you in a second, close to the predicted value of 2. And what's nice about this approach is that in the WBE approach, they assumed that the volume was preserved across all branching generations, so the k plus 10, the volume was the same as the k plus 1. We don't make that assumption, which allows for side branching, which is what plants do a lot. We just assume that the volume downstream of a given branch is proportional to that branch's diameter, which I think is realistic because that branch diameter has to supply that downstream volume. And so looking at this data in the context of subtrees when you're looking at whole networks becomes very important. This also allows branching to follow a Poisson process. So it's not a pure fractal. You develop the expectations for a fractal model, but then it can work for a network that has a characteristic scale like I described before, because we don't have this constraint that the volume's conserved. So it's just proportional to the diameter. Everything downstream is proportional. And so we have these 6 predictions. Let's look at some of the empirical data. There are 4 data sets here. The eucalypt trees that I described, both the raw data and then looking at it as subtrees. The stem data is the Banksia data that I talked about before, where it's the terminal branches of about 180 Banksia species, which had various numbers of, we took the current year's gross, went back to the bud scar, cut everything distal. That's why there are more stems than there are species. Petiole data, this was data I collected while I was at Georgia Tech for about 30 species, 20 to 30 petioles per species. Again, intraspecific relationships, but also we can look at it interspecifically. And then this is a single leaf vein. I've looked at several leaves, but one single leaf vein leaf has over 9,500 veins. The prediction for flow similarity is that length should go to diameter power 2. For the raw data, it doesn't work very well, but if we look at the slope for subtrees, the mean value is very close to the predicted value of 2. We see the same thing in the stem data, petiole data, and leaf veins. Surface area to volume 3/4. Doesn't matter really if we look at raw data or subtrees. We get something close to 3/4. Same thing for stems, petioles, and leaf veins. Now these data sets are biased toward distal parts of the plant network. We're looking at leaf veins, petioles, terminal stems, and small trees. So I don't have a lot of data. I don't know of anybody who's measured the dimensions of the oak tree out there. Nobody does it. But it would be great to know how far we can push this. Do we start to move towards elastic similarity? What happens? When we look at all of the surface area to volume data together, the red lines are the standardized major axis regressions. It's one line has 2 red borders. I did it that way to make it easier to see. They all tend to fall on top of one another, and it's made me wonder whether or not the characteristic scale, which we saw did not vary across leaf size, so maybe there is a characteristic scale or an average characteristic scale for plants, whether that combined with volume conservation leads to convergence along a single function. This is very speculative at this point, but interesting to pursue. You can ignore the math. For 6 bivariate relationships, we have 15 pairwise relationships between their scaling exponents. So this is, again, we're looking at covariation between scaling exponents, like I described before. For example, we can look at how the surface area to volume relationship changes with the length to diameter relationship. We can determine the covariation function for that relationship, and then we can look at that compared to empirical data. These are petiole data. So, for example, surface area to volume against length to diameter. The red line is the predicted covariation function. Each data point is an empirical exponent from a species. So again, 20 to 30 petioles for that species, from the smallest to the largest I could find. Determine the scaling exponent. There's about 30 data points there, something like that. The R-squared value is the sum of the squared deviations from the predicted function. And so it does a fairly good job of capturing the allometric covariation across these different species. Full disclosure, this model is based on a cylinder, so we shouldn't be too surprised when petioles, which are fairly cylindrical, conform well to a cylindrical model. But you have to start somewhere. I don't have data for whole trees. I'd love to see data for whole trees on here. But what I like about this is we can then start to think about, okay, what do different ecological or evolutionary scenarios, where do they put you on these functions? And start to understand the allometric state space in that way. So I've used volume conservation and velocity preservation as model constraints, and I think they're fairly parsimonious model constraints, but I've wondered if the target of selection might be a more integrative trait, and one that has popped out is Sapwood-specific conductivity. So Sapwood-specific conductivity is proportional to flow rate over pressure gradient length over radius squared. So if flow rate and pressure gradient are preserved and length to the radius squared is preserved, that conserves Sapwood-specific conductivity. And there are a few people who have looked at this in trees, at the terminal branches of trees, and in fact it is a species-specific trait. Which reminds me that the model, we've invoked it as a species-specific model. I think the difficulty with going from species-level modeling to interspecific modeling is density changes, and that's a difficult one. So to make that leap, I think you have to understand how density changes and why. But you can use the same model on all of those different species. So in some of the earlier work that I described, plants and animals were actually treated very similarly from a modeling standpoint. The features of the animal model, the Westbrown-Enquist animal model and plant model, there are differences, but a lot of the features of the model are actually very similar. And so it's kind of a one-size-fits-all approach. And I think there are reasons that's not going to work as well for plants, one of which is energy minimization. So for an animal, there's a direct cost in terms of ATP to pump blood through the cardiovascular system. For a plant, water flows via evapotranspiration, this negative pressure gradient through the plant. That's pretty much free. You still have to control the stomates. There's a metabolic cost for that, and you still have to build the network. But I think it's probably not as strong a penalty as it is for animals. And the other thing is that foraging is different. Animals carry their networks around. Plants don't. Plants have to forage via network growth. And I think that's why we have this characteristic scale that keeps popping out, because there's, you can think of roots foraging in soil, or plants, or stems growing to intercept light, that there's some average length scale over which you need to do that, which leads to differences potentially in how these networks are formed. However, there are some sessile animals. Gorgonian coral is one of them, and on a whim I looked at the frequency distribution of its length, and in fact it has a characteristic scale much like that in leaves, and it's solving a similar problem where it has to intercept, these things are filter feeders, it has to intercept nutrients coming by in it, so it fills that space with some average length scale. So I think this idea of a characteristic scale is really underappreciated in the plant network literature, and potentially in a broader scale. So I'd like to finish with a quote from Carl Nicholas. Regardless of their apparent elegance, however, scaling hypotheses derived from first principles are notoriously fragile, often shattering in unexpected ways when confronted by hard data obtained from the real world of biology. So we shouldn't be surprised when our 1, 2, and 3 parameter models don't fail to explain all of the variability in the universe. They're just models. That said, all models are wrong, some models are useful. And this is very much the spirit in which I approach this stuff. These are caricatures, they're abstractions of the way the world works. How far can we take geometry to explain, based on physics, which is important, but how far can we take that geometry based on physics to explain variability in plant morphological and functional data? And then what else becomes important to understand what's out there? I'll acknowledge some collaborators, particularly Josh White, Van Savage, Brian Enquist, Michael Renton. Also Carl Nicholas deserves mention. People who have provided funding. And with that, I'll take any questions.